Welcome to class PM. The previous class we have studied about the components of the cell, right? And we have seen about the nucleus, right? We have spoke about the cytoplasm. See, it could be a unicellular organism or it could be a multicellular organism. Every cell has a layer which is surrounding the cell to keep the components of the cell away from the external environment. At the same time, the cells need a lot of chemical activities. They do a lot of chemical activities, and for which they need a little membrane bound organelles, right, which will help them support. See, like we know that the stomach which is present in our body will help in process of digestion. In the same way, the kidneys which are present, they help us to throw the waste, right? And same way, the lungs will help us to respire. So in the same way, cell also has to perform different activities, and these activities need a lot of chemical support, right? And these are being done with the help of little membrane bound organelles, right, which are seen in every cell. So this is a distinguished feature of a prokaryotic cell and a eukaryotic cell. That is what we have been learning right from the beginning of the chapter, that the prokaryotic cells devoid of membrane bound organelles. So this class we are going to learn about the cell organelles of eukaryotic cells. So these cell organelles are present only in the eukaryotic cells. They are absent in prokaryotic cells. So this class we are going to learn about few cell organelles, and the very first we are going to begin with is endoplasmic reticulum. Endoplasmic reticulum, which is also commonly called as ER. So this endoplasmic reticulum is a large network of membrane bound tubes and sheets. So as I've told you, first we'll learn about the first cell organelle, that is endoplasmic reticulum. This endoplasmic reticulum is present as a large bound organelles of sheets and tubes. You can say a network of membrane bound tubes, network of membrane bound tubes and sheets, and these are also seen as vesicles. They also look like round, or you can say sheets or round or oblong bags. So when you talk about endoplasmic reticulum, they are actually present as a form of network of membrane bound tube-like structures or sheets, and sometimes you can also say that it looks like a round or a oblong bag, a vesicle, a bag, right? Oblong is like which is of not proper shaped, right? And these endoplasmic reticulum which is present inside the cell is classified into two types. You know, it is similar to plasma membrane. The structure of this is similar to plasma membrane, and when you talk about this, ER is broadly classified into two types as SER and RER. SER is nothing but smooth endoplasmic reticulum. RER is rough endoplasmic reticulum. So when you talk, the endoplasmic reticulum which is present inside the cell is broadly classified into two as SER and RER. SER is smooth endoplasmic reticulum and RER is rough endoplasmic reticulum. I'll tell you the difference. The SER, when you look it under microscope or when you observe it under microscope, the surface of SER is smooth. It is smooth. But whereas when you look at these rough endoplasmic reticulums under the microscope, they seem to have what you call ribosomes present on it. They are just like oblong vesicles, right? Like this. When you look at the structure, right, I'm just showing a normal, this is not the exact thing, but they have ribosomes which are present. They are oblong structures, vesicles, right? So you can see the ribosomes which are present on the surface of RER, due to which it seems to be rough. That is the reason this is said to be a rough endoplasmic reticulum. So this RER which is present, it is said to be rough endoplasmic reticulum because they have ribosomes on their surface, right? And these ribosomes which are present, these are actually ribosomes are sites of protein manufacture. So that means these endoplasmic reticulum, RER, which is present, it synthesizes protein in the cell. So as they have ribosomes, always these ribosomes are concerned with the synthesis of proteins. But you see that this SER which is present, it is always, SER is always a site of fat manufacture or lipid manufacture. So they are always in the work of manufacturing or synthesizing fats in the body. So the fat and the protein which are manufactured in the cell has to be transported to different parts of the cell, which is done through these long tubes or sheets, which is said to be endoplasmic reticulum. So you might have understood that this ER helps in transportation of proteins and lipids which are synthesized within the cell. Okay, as I've told you, the ribosomes are naturally the sites of protein manufactures, and this SER which is present, it synthesizes lipids or fats which are needed for the cell, right? And these proteins and lipids which are manufactured are transported to the different parts of the cell through the ER. And see, the cell uses up these proteins and lipids. The cell uses up this proteins and lipids, and the excess proteins and lipids which are present, they use it up for building the cell membrane. They use it up for building the cell membrane, and that process is said to be biogenesis. What is it called? Biogenesis. I'll tell you what is it. Okay. So this biogenesis is nothing but building up the cell membrane using proteins and lipids. Usually this biogenesis in class 9 is asked for 1 mark. Okay, it's an important question. So whatever synthesis, like how much ever they synthesize, like protein or lipids in the cell, they usually transfer it to different parts of the cell body. But they also use up some of these protein and lipids to build up the cell membrane. And this process of building up the cell membrane using these proteins and lipids is said to be what? Biogenesis. It is said to be biogenesis. So we know that few proteins and lipids are used up for the building up of cell wall. At the same time, few proteins and lipids act as enzymes and hormones. Though the ER varies in different cells, the function is one and same. That is, they help in transportation of the material in the form which is carried in the form of long channels or tubes, right? It could be transportation of these materials between the cell and the cytoplasm, or between the cytoplasm and the nucleus. And in few cases, they also, eukaryotes, they also help in, like we are talking about the eukaryotes only, right? In case of few cells, they also carry out some biochemical activities. Biochemical activities where liver cells, in case of few invertebrates, there are few toxins which enter, right? This ER which is present, they detoxify those. In case of vertebrates, you know what vertebrates are? Vertebrates are the organisms which possess backbone, right? So in few of the vertebrates, this SER which is present, it will help in detoxifying the poison or detoxifying the chemicals which enter into the liver cells, That means they play a crucial role, important role. So this is all about your endoplasmic reticulum, where it will help in transportation of the materials. At the same time, in few vertebrates, it will also help in detoxifying, and few of the proteins and lipids which are synthesized will act as enzymes and hormones. So now let us learn the next cell organelle, that is Golgi complex. So the next cell organelle you are going to learn is Golgi complex or Golgi apparatus. You can also call it as apparatus. So this Golgi complex which is present, it is actually first described by a scientist called Camillo Golgi. It was first described by a scientist called Camillo Golgi. It is a membrane bound organelle. So when you talk about this Golgi complex, for the first time Camillo Golgi was a scientist who observed this Golgi complex as you can say cisterns, a pack of tubes or pack of sheets running parallel to each other, which are said to be cisterns, So you can see membrane bound vesicles arranged parallelly to each other called to be cisterns. So you see that this Golgi complex, okay, These Golgi complex are membrane bound vesicles or sheets or tubes which are arranged parallel to each other, and this packed tubes is said to be a cistern. It is said to be a cistern, and the membranes often, these membranes, Golgi complex or Golgi apparatus, they often have a connection with ER, that is endoplasmic reticulum. We know that the function of the endoplasmic reticulum is to synthesize lipid and proteins and transport it to different parts of the cell. In the same way, the Golgi complex which is present, it is mainly involved in synthesis of, in transporting the protein and lipid which is synthesized by ER. That means the ER synthesizes the protein, or it makes the protein and lipid, and they pack it and dispatch it to different components of the cell within the cell and outside the cell through this Golgi complex. That means they act as drivers which drive the lipids and which drive the proteins to different parts of the cell body, at the same time outside the cell body also. So that means the packing of the food and dispatching of the food is done by Golgi complex. That is because these membranes are connected to ER. These membranes are connected to ER. So ER will synthesize and this will pack the food and dispatch it to the different parts. Just like a factory which will prepare chips, right? You can say, like, let us take example of a lace factory. What do? They synthesize. That means they make the lace, right? And they pack it, dispatch it to the various places or various cities in the country so that you can get it, right? In the same way, though our cell is a small city, it has small structures which will help in packing the food and dispatching the food. So what are those small substances which will help in packing the material? That is Golgi complex, and they also help in dispatching. And you know that the main function of this Golgi complex is to storage, modification, package of products and vesicles. So the main function of this Golgi complex is storage, storage, modification, if needed they modify, package of the products, that is the proteins and lipids or whatever, in vesicles, because they are always connected to ER. The membranes are always connected to ER. So all the time, as they have a good connection and good contact between each other, they try to send the food in the form of packed structures. And you know, this Golgi complex also help in formation of lysosomes. Help in formation of lysosomes. You'll be learning about these lysosomes. They help in the formation of lysosomes, right? So this is all about the Golgi complex, that is the next cell organelle. The next cell organelle. So we have seen that the Golgi complex help in storage and at the same time dispatch of the products which are formed inside the cell. That is, with the help of ER, they help in transportation. Right? We also have seen that the Golgi complex helps in formation of lysosome. So the next cell organelle which you are going to learn is lysosomes. Lysosomes. These lysosomes are the membrane bound organelles which are present. And you can say that, as I've told you, the cell carries out many activities, right? It will take in substances, it will give out substances. Giving out substances is what? It is nothing but throwing out the waste, right? So these lysosomes are a little related, like they are related to disposal of waste out of the cell. That means you can say that they are always in the process of disposal of waste out of this disposal system. They form the disposal system of cell. That means they are always concerned or always related to throw the waste out of the cell which is produced during various metabolic activities. So they form the disposal system of the cell. As I've told you, these lysosomes form the disposal system of the cell. See, usually these lysosomes are help in the digesting the substances inside the cell. Say, like, if any foreign bacteria enters, or any bacteria or any germ enters into the cell, the lysosomes, they have powerful enzymes. And what do? They kill this bacteria which is entering into the cell and break it into small pieces. Break it into small pieces so that it can digest it. So during this process, if there is a disturbance in the metabolic activity, say like if the cell gets damaged, the lysosomes, what do? They release the enzymes, break the cell into pieces, and they dissolve the cell. They eat up the cell by themselves. Or you can say that they burst the cell. They burst the cell and enzymes will digest the own cell. The enzymes which are released by the lysosomes will digest the own cell. That is the reason these are called as suicidal bags of the cell. What are these called? Suicidal bags of the cell. So let me show you. Say if this is the cell, right? If this is the cell, this is the nucleus. Say like if some foreign bacteria enters into the cell, the lysosomes which are present, they digest the bacteria which is entering by releasing the enzymes. They break up the bacteria into small pieces, and during this metabolic activities, if the cell gets damaged, the enzymes will burst out, okay, which are the lysosomes which are present, they burst and release the enzymes so that they can digest their own cell. So by doing this, that means they are committing suicide, they are killing themselves, right? So that is the reason they are called as suicidal bags of the cell. Or you can say that the enzymes which are present inside the lysosomes are made by RER. Enzymes are made by RER. So the enzymes which are present, they are made by RER. And you need to remember that these digestive enzymes are very powerful. That's the reason they can break down the bacteria or any foreign germ which is entering into the cell, okay, into small pieces. If in case there is a damage caused to the cell, these enzymes the lysosomes will burst, the enzymes will digest their own cell and they kill themselves. That's the reason they are called as suicidal bags. So this is all about your lysosomes. Now let us continue with the next cell organelle, that is mitochondria. Mitochondria. Yes. Now when you talk about this mitochondria is another membrane bound organelle which is present within the cell. And you know, this mitochondria is known as powerhouse of the cell. What is it called? They are called as powerhouses of the cell. They are called as powerhouses of the cell. Okay. The mitochondria are called as powerhouses of the cell. I'll tell you why. We know that to do various activities right from the beginning of the day till the end of the day, we need a lot of energy, right? So from where do we get energy? From the food which we eat. It is not only that we feel that we are working, right? But at the same time, the cells which are present in our body also continuously work. They continuously work. And when they are continuously working, they need to have energy in sufficient amounts so that they can carry out the activities. So this energy which is needed to carry out various metabolic activities in the cell is provided by mitochondria. So this, do you know how do they provide this energy? I'll tell you. The energy is provided, the energy is released in the form of ATP. I'll tell you what does this ATP stand for. ATP is nothing but adenosine triphosphate. So as I've told you, this energy is released in the form of ATP. And this ATP is nothing but adenosine triphosphate. You know what is this ATP is called? The ATP is called as currency of the cell. It is called as currency of the cell. So this currency which is present, the power which is present in the ATP is released into the cell so that it can carry out mechanical activities or metabolic activities. And you know, the surface of this mitochondria has two layers, inner membrane and outer membrane. So when you look mitochondria, this is the outer membrane, okay, the outer membrane which is present, and the inner membrane is just thrown into folds like this. Okay, it is thrown into folds like this so that these folds which are present, they provide a surface area for ATP reactions, that is energy currency reactions. Okay, you can say that they provide a larger surface area for ATP generating chemical reactions. So this is what the structure of mitochondria, when you observe it under microscope, you see that they have an outer membrane which is a normal membrane, but whereas the inner membrane is thrown into several folds which will provide a larger surface area for ATP generation reactions. So we know that ATP is the important component which will provide energy for the cell. And you know, this mitochondria is considered to be a strange organelle, You can say that it has its own ribosome, so it can synthesize its own protein. It will not take protein from any other substances or any other organelle. It can synthesize its own protein. They have their own DNA, right? They also have their own DNA. So that's the reason they are called as strange organelles. As mitochondria has their own DNA and ribosomes, they don't depend upon any other organelles for their energy or for their food. Because when there is ribosome, we all know that ribosome is a site of protein manufacture, right? So when mitochondria can synthesize its own protein, why does it has to go and depend upon any other organelle, right? So that means it is independent, it is not dependent. So that is the reason it is called as a strange organelle. That is the reason it is called as a strange organelle. And always remember, this mitochondria is said to be the powerhouse of the cell, and the power is released in the form of ATP. That's the reason the ATP is called as currency of the cell. We know that various countries have different currencies, right? So the currency of the country cell is nothing but ATP. Okay, and if you remember, we've been learning that during the process of photosynthesis, along with the glucose and oxygen, energy is given out, right? The energy which is given out during the process of photosynthesis is also released in the form of ATP. So, you know how many ATPs are released during the process of photosynthesis? It is 38 ATP molecules. 38 ATP molecules. And these ATP molecules which are released during the process of photosynthesis in the plant cell are utilized to carry out various metabolic activities. So, the next cell organelle which we are going to learn is plastids. And these cell organelles are usually seen only in plant cells. And you know, these chloroplasts which are present, when you look at a plant cell under the microscope, you find cells which are plastids. There are plastids present which have chloroplast in them. And there are two types of plastids. Okay, there are two types of plastids. Plastids are categorized into two types as leucoplasts and chromoplast. Leucoplasts and chromoplasts. And these chromoplasts are colored plastids. They are colored, and these are colorless. You can say white. We generally don't say white, but they are colorless plastids. They are colorless plastids. So when you talk about these plastids are absent in animal cells, because animals do not have any pigment called chlorophyll, right? We only know that plants have chlorophyll in them, where these chloroplasts which are present, they help in promoting the color to the leaf, right? And these plastids which are present, they are found only in plant cells, and they are of two different types. These plastids are of two different types, like leucoplasts and chromoplasts. Leucoplasts are usually colorless, or we generally call it as white, and chromoplasts are usually colored. They can be in plastids, they contain chlorophyll. We know that chlorophyll is a pigment, and these chromoplasts are usually yellow or red in color. That's the reason you see different fruits of different colors, right? See, when you take carrot, we know that carrot is orange in color. How is this orange color being given to the carrot? That is because of a pigment chromoplast, which is nothing but a carotene. Carotene is a pigment which will impart the color to the carrot, right? Impart the color to the carrot. And when you talk about, say, some other color, like sometimes we see that there are leaves which are yellow in color, right? There are leaves which are yellow in color, or flowers which are yellow in color. That is because they have a pigment chromoplast, right, which is of yellow in color. So that is how the color is being imparted to fruits or flowers, right? In the same way, as I've told you, the leucoplasts, they are colorless. You cannot distinguish any color. But whereas the chlorophyll pigment which is present, it will have chloroplasts in them, right, which will help in the process of photosynthesis. You can see the photosynthesis is carried out by the chlorophyll, presence of chlorophyll. And these leucoplasts which are present, they store starch, oil and protein granules. So these leucoplasts which are present, they usually store starch, oil and protein granules which are present. And so when you talk about the plastids, as I've told you, they are usually found in plant cells, and these are of two types called leucoplast and chromoplast. So the leucoplasts are colorless or white, and the chromoplasts are usually colored, and they can be seen in yellow or orange color, right? As I've told you, keratin is a pigment which will promote the orange color to the carrot, right? They also contain chlorophyll, and these chlorophyll are present inside the chloroplast cell. When you look at the internal structure of a chloroplast cell, this is how the cell of chloroplast is, right? And you see that these are the stacks, they are present as a stacks, and these stacks are called as grana, and these grana are connected to each other. Okay, you can see disc-like structures, right? These are said to be the grana which are present in the chloroplast, and these are connected to each other through a long tube-like structures called as stroma. What are these tube-like structures called? Stroma. When you look at a chloroplast cell under the microscope, you can find there are structures called grana which are present as disc-like structures, as they're stacked one upon each other. They're placed one upon each other or piled one upon each other, where they're said to be stacks of grana. Stacks of grana, and these are connected to each other. There is a connection between these stacks through a long tubular structure called as stroma lamellae. Stroma lamellae. So when you look at the internal structure, this is how the chloroplast cell seems to be, and we know that this chlorophyll or the chloroplast which is having a chlorophyll pigment will help in the synthesis of food, that is in case of plants. And these plastids are just like mitochondria. They are independent. That means they don't depend upon any other cell organelle for synthesis of their food. That means they have their own DNA and ribosomes. They have their own DNA and ribosomes. When it comes to the ribosomes, immediately it should strike in your mind that ribosomes are the sites for manufacture of protein. So we know that protein is needed for building up the body, right? So if the cell has to build up, then definitely they need proteins, right? And these proteins are synthesized within the cell of plastids. So they don't need the protein to be transported from different parts of the cell. They can synthesize their own proteins. So this is all about the plastids. Not the least, the next cell organelle are vacuoles. The next cell organelles are vacuoles. So when you talk about these vacuoles, these are actually the storage sacs. What are they called? Storage sac means a bag, right? So storage sacs for food and water, or you can say for solid or liquid, for solid or liquid. So these are usually the storage sacs. They are called as storage sacs because they store solids or liquids inside the cell. Solids or liquids inside the cells. And the animals, in case of animal cell, the size of the vacuoles are very small. The size of the vacuoles are very small. But whereas in case of plant cell, you have large sized vacuoles. When you observe it under microscope, you see that there are large sized vacuoles which are present in case of plant cell. But whereas in case of animal cell, there are very small but numerous in number. And the central vacuole of some plants, okay, the central vacuole or the central storage sac of the few plant cells has usually occupy 50 to 90% of the cell volume. If this is the cell of a plant, so you can see that in case of few plant cells, the vacuoles occupy almost 50 to 90% of the cell, 50 to 90% of the cell volume. And these plant cells, the plant cell vacuoles are filled with a cell sac or full of cell sac. Assume that this is a plant cell and it has a vacuole in it. I told you that in case of plant cells, the vacuoles are large sized, right? So these large sized vacuoles will have cell sap in it, which will provide the turgidity to the plant cell or rigidity to the cell. Okay, the cell sap provides turgidity and rigidity to the cell. That is nothing but toughness, turgidity and rigidity to the cell. So you can say that the cell sap which is present in case of most of the plant cells, they provide the turgidity. That is the reason when you try to bend a branch or when you try to pluck a leaf, they are very strong because the cells have the turgidity That they have the rigidity because of the presence of cell sap. And you know, the cell vacuoles which are present, the vacuoles which are present in case of plants, they store many important things of life, like amino acids, like proteins, like sugars, right? And you can say food, right? In case of amoeba, the vacuoles store food. The vacuoles store food. And in case of unicellular organisms, as I've told you, these are the storage places for food, right? Solids or liquids. So in case of most of the plant cells, they store many important things like amino acids, sugars, proteins. And when you talk about a amoeba, they store food inside the vacuole. And in few unicellular organisms, we know that unicellular organisms only process their life activities through single cell, right? So in case of such unicellular organisms, they throw the waste out of the body, expel the excess water out of the body through the vacuoles, That means some waste from the cell is given out through the vacuoles. That means they also play important role in expelling excess water and waste. So this is about the vacuoles and the cell organelles of a cell.